This short biology lesson will look at the structure of a DNA molecule. DNA stands for deoxyribonucleic acid. Nucleic acids are a family of macromolecules, large complex molecules that we find in living things. There are two main types of nucleic acid. One, of course, is deoxyribonucleic acid, or DNA. The other is ribonucleic acid, RNA. Today we'll just look at the structure of DNA. DNA is sometimes referred to as the blueprint of life, and this is because it contains a set of instructions that code for proteins that enable a cell to function normally. It's also a molecule that needs to be copied when a cell or a living organism is propagating itself. That set of instructions needs to be passed on to the next generation so that it too can function normally and as it should. The structure of DNA forms a double helix structure consisting of two complementary strands of DNA. On the right we can see a model of a molecule of DNA. You can see it's sometimes referred to as a twisted ladder structure. The outer backbone of the ladder, shown in the red and the grey, this is the sugar phosphate backbone. In the next slide we'll have a look at a diagram to break that down. And the blue components in the centre, or the rungs of that ladder, these are the bases of DNA, and it's the bases that are holding the two individual strands of DNA together, forming this double helix. Given that it's a double helix structure, the double helix is composed of two separate strands of DNA held together, as we said, by these complementary base pairing between the bases of each strand. Each strand of DNA is a polymer being a large molecule made up of repeating subunits that are chemically bonded together. In the case of DNA, the subunits that make up DNA are called nucleotides. Looking at the structure of one nucleotide, there are three main components. Each nucleotide must contain a phosphate group. The phosphate group is negatively charged, and this enables DNA strands to be separated in genetic engineering manipulation, and we'll discuss that in a later tutorial. The phosphate group is bonded to a 5-carbon sugar ring. Each one of these points corresponds to a carbon atom. The sugar in DNA is deoxyribose. This sugar molecule is bonded to the nitrogenous base. In this case, the base shown is cytosine. There are 4 bases. So these three components, the phosphate, the sugar, and the nitrogenous base, collectively make up a nucleotide. And it's the joining together of nucleotides that forms one strand of DNA polymer. This diagram shows a short section of DNA that shows two strands of DNA bonded together by complementary base pairing. There are 4 bases in DNA. They are adenine, thymine, cytosine, and guanine. Now, we talked about the double helix structure of DNA having a sugar phosphate backbone. If we have a look at the joining of these nucleotides together, they bond together in such a way that the phosphate and sugar molecules align the backbone of the double helix molecule, as you can see here. So phosphate, sugar, etc., as each new nucleotide is formed onto that DNA strand. You might notice that the strands have different directions, so this strand looks upside down compared to this strand, and we can see that the phosphate group is in a different orientation compared to this strand over here. This is referred to as the anti-parallel nature of DNA. The rungs of this ladder, as we said earlier, made up of the bases that complementary base pair. What we mean by complementary base pair is that these 4 bases will always pair up in exactly the same way. If we have an adenine in this nucleotide, it will base pair only with a thymine nucleotide base, and it will do so by forming 2 hydrogen bonds. If we have a guanine, it will form 3 hydrogen bonds with a cytosine. DNA molecules will always obey these base pairing rules. In other words, adenine will only ever bind to thymine, guanine to cytosine, thymine to adenine, cytosine to guanine. The hydrogen bonding holding these two strands of DNA forms a very important feature of DNA. First of all, being forces of attraction between these strands of DNA, the hydrogen bonds need to be strong enough to maintain the integrity of the DNA helix and preserve that structure. DNA as a molecule is quite stable compared to RNA, which is single-stranded. So these hydrogen bonds need to, under normal conditions, be held together to preserve the integrity of the DNA molecule. On the other hand, those hydrogen bonds need to be weak enough such that they can be broken to expose those bases so that the DNA code can be read in order to make proteins during protein synthesis, and also so that the DNA double helix itself can be replicated prior to cell division. Looking at the organisation of DNA, DNA is, if we're talking about a eukaryotic cell, and of course even a simple bacterium will have a simple circular chromosome of DNA, but if we're talking about the more complex eukaryotic cells such as those that we find in the human body, we have 46 linear chromosomes in the case of humans. Now that covers a fair amount of nucleic acid, a fair amount of DNA that needs to be contained in a relatively small space in the nucleus of the cell. For this reason, the structure of DNA needs to be compacted so that it can be contained in that cell nucleus. Looking at the organisation of the DNA double helix, we can see that, so here we've got our twisted ladder structure, and that double helix is wound around proteins called histones as a way of organising it, and those histones wrap around each other to form a complex called the nucleosome. Nucleosomes then form a complex called chromatin. Now, chromatin fibre is what we think of as the main structure of DNA. Chromatin we tend to associate with the uncondensed form of DNA, and normally if you look down cells under a microscope that are stained, where the DNA has been stained, you won't see the characteristic shape of these condensed chromosomes that we often think of as chromosomes. What we're looking at is the uncondensed form of chromatin, which is just the long chromatin fibres. In preparation for cell division, DNA must be compacted and condensed even further into the structure that we see here as a mitotic chromosome. Now, this is because during cell division, long strands of chromatin can easily become tangled up. They don't separate, strands of DNA won't separate very easily in this particular structural format. So the chromatin condenses, and what we can see here is actually two strands of DNA, two sister chromatids. So one chromosome has made a copy of itself, and those two copies are held together at the centromere region. This condensed chromosome is the structure that we see associated with mitosis, where the sister chromatids are separated in the process of cell division. This diagram elegantly shows the difference between the uncondensed chromatin and the mitotic chromosomes that we see associated with mitosis. These particular cells, these are a HeLa cell line, originally taken from or developed from a cervical cancer patient, Henrietta Lacks, in the United States in the 1950s, and now a very widely used cell line. These two cells on the right, we can see that the DNA, the DNA has been stained with a fluorescent dye so that under a particular wavelength of light the DNA will fluoresce. And we can see that the DNA in this format is largely uncondensed, and so we can't actually see distinct chromosomes that we recognise during mitosis. So this is uncondensed chromatin. This cell here on the left, we can see that the DNA is much more condensed. This cell is in the process of undergoing mitosis, so the replicated chromosomes are dividing to form two new cells. So we can see that the structure of DNA in this cell is largely different to the structure of DNA in this cell, even though the content of the DNA itself is the same.